Lesson 3H, Metabolism. Metabolism is all the chemical reactions that occur in a cell or an organism to maintain a living state. Of a human's total energy use, around 75% is used simply to power the basic resting functions of your organs. There are two broad categories of metabolic reactions. Catabolic, where substances are broken down into products, also known as a degradation or decomposition reaction. Substrate A into products B and C. And anabolic, where substances are combined to create products. We call this the synthesis reaction. Substrates A and B into product C. We call the ingredients of a reaction the substrates, and the material produced the products. All chemical reactions require an initial input of energy to start the chemical reactions. We call this the energy of activation. The human body is not warm enough to meet the energy requirements to drive most chemical reactions, so ATP is used. However, the amount of nutrients, especially carbohydrates, that would be required to run all of the organism's chemical reactions would be so demanding it would likely not be fulfilled even if the organism constantly ate from an unlimited supply of nutrients. So enzymes are necessary. An enzyme is a biological catalyst made of protein, which is something that lowers the activation energy of a chemical reaction without being used up in the chemical reaction itself. The reaction requires less ATP. This graph shows how much energy is required to complete the chemical reaction without a catalyst, and the amount of energy required with the help of an enzyme catalyst. As you can see, an enzyme can significantly lower the energy of activation for a given chemical reaction. Here is an illustration of a catabolic enzyme in blue, degrading a substrate into two products. It does so by lowering the activation energy of the reaction. We have been calling this reaction a hydrolysis reaction. The enzyme will then release the products and be available to bind another substrate, and repeat the reaction again and again. It's important to understand that the enzyme is specific to a substrate and will not bind other substances. We call the binding site the active site of the protein enzyme. In this illustration, we see a synthesis reaction, where substrates bind to the active site of an enzyme. We call this union the enzyme-substrate complex. Now the enzyme will assist in combining the substrates into a product by lowering the activation energy. The enzyme will then release the products and it will be available to bind another substrate. However, the enzyme will not last forever, as it's organic, and it will begin to lose effectiveness with time. As noted earlier, enzymes are specific to a substrate. We call this the lock and key model of enzyme function. A specific substrate acts as a key that fits the matching keyhole of the enzyme, called the active site. Here you can see a model of an enzyme with a specific substrate key in its active site keyhole. The shape of both the protein and the substrate must match for the enzyme to work. Another model that works in concert with the lock and key model is the Induced Fit Model. This model states that the protein enzymes are slightly flexible, and that allows for the enzyme to form around the substrate when it binds to the enzyme, which means the substrate is securely held in place for the chemical reaction to occur. These two models result in the enzyme securely holding the proper substrates in the active site, which we now call the enzyme-substrate complex. This perfect alignment of the substrates in the enzyme ensures that the chemical reaction can occur with a lower energy of activation. All of an organism's chemical reactions occur with the help of enzymes. If a chemical reaction is required, the cell or organism will manufacture the protein enzyme, and the rate of the chemical reaction will increase. As the enzyme ages, it will degrade itself, and the rate of the specific reaction will slow down. If the organism senses a need to increase the specific reaction again, it will produce more enzyme.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.